Friends, today is our topic TCA cycle or tricarboxylic acid cycle, which was discovered by kreb. That's why known as kreb cycle. It's occur in mitochondrial matrix. First thing we discuss in it is regulation. It's regulated by phosphodehydrogenase complex enzyme, dehydrogenase complex enzyme, which consists of 5 coenzymes: E1 pyruvate decarboxylase, E2 dihydrolipolyl decarboxylase, transacetylase, E3 dihydrolipolyl dehydrogenase, and pyruvate dehydrogenase kinase phosphorylate, which inactive E1, and pyruvate dehydrogenase phosphate dephosphorylate, which activate E1. So there we discuss first regulation of tricarboxylic acid cycle, which caused by pyruvate dehydrogenase complex, which consists of 5 enzymes, which I written in below. You see it. So it's occur in inner mitochondrial membrane. First, it's discovered by kreb. Basically, it is series of chemical reaction like glycolysis, in which pyruvate is oxidized into carbon dioxide. So it's also like glycolysis give us energy for for different cellular functions. So first, the pyruvate obtained from glycolysis is converted into acetyl CoA. Pyruvate is 3 carbon compound, which converted into acetyl CoA, which is 2 carbon compound. So when pyruvate converted into acetyl CoA, there we gain one NADH and one carbon dioxide molecule, and this cycle go on forward. Then acetyl CoA is combined with oxaloacetate, which is 4 carbon compound. Now acetyl CoA and oxaloacetate combine to form citrate, which is 6 carbon compound. Oxaloacetate is 4 carbon compound, acetyl CoA is 2 carbon compound, which both together converted into citrate, which is 6 carbon compound. Then citrate converted into isocitrate, converted into alpha ketoglutarate, and there we obtained another NADH and one carbon dioxide molecule. Alpha ketoglutarate is 5 carbon compound. Then alpha ketoglutarate converted into succinyl CoA, and there we gain another NADH and one carbon dioxide molecule. And succinyl CoA converted into succinate, which is 4 carbon compound. There we gain one GTP molecule. And FADH, then succinate converted into fumarate. There we gain another FADH too. Then fumarate converted into malate. Malate converted into oxaloacetate. We again gain oxaloacetate. This is simple. It's all about tricarboxylic acid cycle. In previous lecture, we discussed about glycolysis. I forget that told you about what is the rate limiting step of glycolysis. When fructose 6 phosphate, which is third step of glycolysis, it's rate limiting step. It's stimulated by phosphofructokinase 1. Phosphofructokinase 1 is stimulated by ADP and inhibited by ATP. So it's the rate limiting step of third step of glycolysis. It's called rate limiting steps, because if this step is inhibited by ATP, so the glycolysis is stopped. It's not occur in cell. If you stimulate, then it's occur. So that's why it's called rate limiting step, because it decide whether glycolysis occur or not. And then we discuss glucose transporter proteins. So one these are in 1 to 14 in number. Glucose transporter protein 3 transport glucose in nerve cells. Glucose transporter protein 4 transport glucose in muscle cells. Glucose transporter protein 1 transport glucose in blood. Glucose transporter protein 2 transport glucose in kidney and liver. So it is our today lecture. I hope you like it.